In this video, we're going to look at experiments. We know that scientists learn new information about the natural world by performing investigations, which can be done in many different ways. Some of those investigations involve simply observing something that occurs and recording observations. However, some are more involved, and they involve experiments. These experiments are building a model that represents something in the natural world, and then we can use that to test and see how it reacts with different changes. On the screen is the flowchart from the textbook. Now, we know that not all science investigations require an experiment. And when you go through our list here of steps, such as observing a phenomenon and then posing a question about that, we can think about what comes first, the observation or the question. But as you can see, this gives us sort of a stepwise look at what could possibly happen in an experiment. So if you have a question and you want to have an answer to that, you form the hypothesis. And after you have the hypothesis, you can set up an example or a test in the model to see whether or not it reacts the way we expected in the natural world. So here's some basics of an experiment. First is the hypothesis. That's our proposed explanation for a problem. Okay, so in a scientific experiment, the scientists would want to answer their question they've posed, and that's the hypothesis. And the second thing, we have to collect data. So to do this, you create a test, and you change one thing that you'd like to know about the phenomenon, and you do it in a controlled situation, and that one thing you change is the variable. You want to change one variable to see how it impacts other variables, and we'll talk about that in the next slide. So the two main variables we want to talk about in an experiment are the independent variable and dependent variable. Now all variables are changes in experiments. Now if you look on the left hand side, the independent variable, those are the changes made by the scientist to see the other changes. Right? And that's going to change the dependent variable. It's also known as the experimental variable. So as a scientist, you're the one that's setting up this variable. So if you wanted to test, say, the impact of light on light fabric as opposed to dark fabric, right? Your difference in fabric is the test you're making. In medicine, this would be the new treatment you're trying on somebody. So that could be also known as the treatment. On the right-hand side, we've got the dependent variable. This is changes due to an experiment. And this is going to change due to the fact that we've created this independent variable. We also known as the responding variable. And these are our observations during the experiment. These are our results. This is the information that we are collecting. So let's look at an example experiment and apply these terms that we just learned. You can read along as I read aloud to you from the slide. Sam and Marie are investigating plant growth. They know that plants need sunlight, water, and nutrient to grow big and strong. They think that the type of fertilizer will be a big impact on the growth of their plants. They set up the experiment with 3 different types of fertilizer and observed the height of their plants over 2 weeks. So we want to know is what would be Sam and Maria's independent variable? And it's somewhere on the table. Remember, an independent variable set up by our experimenter, right, in this case Sam and Maria, at the beginning of their experiment. It's the treatment, if it were medicine, right? And it's also what we would call our test. So remember, on their hypothesis, they felt that different fertilizers may impact the growth of their plants. So it's the fertilizer that is our independent variable. Fertilizers A, B, and C. Now note at the bottom, D does not receive the treatment, so it would not be considered part of the independent variable. Now, next slide here shows us dependent variable. So this would be the responding variable. This is going to be the thing that's going to change as a result of our experiment. So we're experimenting on plants. We gave them a treatment, which is our independent variable, and now we know what happens. Remember, this is the data we collect to know that are being produced by our experiment. These are the observations we make during the course of our experiment. So on this next slide, we'll see what is the dependent variable. As you can see, the height after 2 weeks is the dependent variable because it's changing as a result of our experiment. This brings us to the next two important terms when we talk about experiments. Experiments are controlled environments where we want to know what one thing we've changed impacts the particular phenomenon that we're studying. And in order to do that, we had to have constants in place, right? Fact that does not change whether the other variables change. So, a control in medicine, we call it a placebo, so we can compare how things are changing, right? So the question is, how do the constants and controls impact a plant test? So here's our data again. Can you identify the control in Sam and Maria's experiment? Remember, a control is something that we set aside. It's a placebo. We don't want it to change during the course of our experiment, so we can compare the other changes. As you can see, plant D is our control because it grew without the benefit of fertilizer, but it also received the same amount of water and sun as the other plants. So we're comparing it as if, that was our control. Then we also want to know what constants we had in place for Sam and Maria's experiment. So this is something that all the plants would have received, regardless of whether or not they had the treatment or the different types of treatment. As you can see, the controls in our experiment, the values that were unchanged, include the amount of water and the amount of sun, because those were the same for every single plant. And that's one of the things about having a controlled experiment. These constants do not change, so we can have a good comparison between what's happening with our treatment and not with our treatment. And the last thing we want to think about is our data itself, right? We got some information from the end of our experiment in our dependent variable. Right, so what could we do to analyze Sam and Maria's data? What observations can we get from the experiment? So on the screen there, I have written some things. So if we compare everything to our plant D, that was our control, we can see that the median plant growth, right, so median being the middle number, 15 centimeters, and the mean was 16.5 centimeters. So we can see that the smallest plant was the one without fertilizer, which is what we would expect if our hypothesis were to be accepted. And we can see that plant C was 40% bigger than the control. So all the plants with fertilizer grew larger, but fertilizer C may have had a bigger impact on plant growth. So whenever we want to write a conclusion, we want to write some type of statement that we would reflect back on the data we collected. So an example could be fertilizer C produced 40% larger plants. And every time you're writing a conclusion, you always want to relate it back to your hypothesis. Fertilizer did impact plant growth, and you should always adjust how you can move on. Changing the amount of fertilizer C may reveal how much is needed. So in review, experiments require us to think about a control, because we want to compare it to something. In this case, with the plants, it was plant D. You want to make sure that you keep all of your constants the same for all of your subjects, whether they got the treatment or not, so that you have a good comparison. And finally, your independent variable, you only want to pick one, so you know that that one variable is making all of the changes, and that your dependent variable is your measured results, and you want to use that to do your data analysis. This concludes this PowerPoint presentation.